spectrum. Okay, looks like the recording's working. Um, both these early and later clinical manifestations reflect, importantly, injury to specific segments of the basal ganglia, the globus pallidus and subthalamic nucleus, as well as auditory brainstem nuclei, the cerebellum, oculomotor nucleus, and hippocampus. Kernicterus also has a characteristic neuroimaging pattern on magnetic resonance imaging, including both the early T1 weighted and later T2 weighted abnormalities, again in the globus pallidus and subthalamic nucleus, occasionally noted on MRI of hippocampus as well. And finally, Kernicterus has a very typical postmortem neuropathology, again, being primarily a neuronal injury, again, in these same regions that highlighted in terms of the neurologic phenotype and the neuroimaging. The only prerequisite condition is a neurotoxic bilirubin exposure. And I think this much we understand, and I also think this much we can agree upon. Notably, the neurologic phenotype, the neuroimaging on MRI, and the neuropathology are entirely congruent, and they validate each other. But what do these consistently reproducible findings validate? Well, they validate a very distinct pattern of central nervous system vulnerability, highly localized bilirubin-induced injury. A topography, I would assert, we don't fully understand. And one of my goals this morning is to highlight this distinctly regional Kernicterus topography and speculate what it might tell us about bilirubin-induced injury, and suggest that an accounting for this localized injury is required for it to gain conceptual clarity of Kernicterus pathogenesis. I would also assert that and highlight that our understanding of the pathogenesis of Kernicterus at the cellular and molecular level is riddled with complexity and remains far from settled. And this, again, this is not for a lack of data. Indeed, we're awash in data, but I think reflects the limited integration of data across a range of biological scales. And in particular, from the molecular and cellular levels to broader cellular networks, neural networks, and whole brain function. And that there is, in fact, a large translational gap between these reductionist findings and what we see or know or understand in vivo. But also, I want to highlight that there's really been very limited differentiation of bilirubin neurotoxicity as a function of time. And here, in terms of distinguishing the very early proximal inciting molecular events and later secondary responses. Remembering that the term genesis in the word pathogenesis refers to beginning or coming into being. So one of my other goals this morning is to highlight the need to define more clearly the temporal sequence of events that comprise a CNS bilirubin injury cascade. And ultimately, with the goal of establishing a timeline of bilirubin-induced injury, with a focus on early events, albeit understanding that later events may very well be important. These secondary responses, including neuroinflammation, may play a cytotoxic or neuroprotective role and potentially contribute to injury, or protect against it, and perhaps at times being cytoprotective and at other times being, cytotoxic and at other times being neuroprotective. Well, let's talk about the CNS topography in Kernicterus. And again, already highlighted the subcortical nature of the injury and the specific involvement of the globus pallidus and subthalamic nucleus, cochlear nucleus, oculomotor nucleus, Purkinje cells and dentate nucleus of the cerebellum, and the metabolic sector of the hippocampus. Now, this very selective involvement, importantly, is clinically evident very early on, essentially right from the get-go. in the initial phases of injury that ultimately lead to damage. And here I'm referring to, of course, the advanced stages of acute bilirubin encephalopathy, where aberrations of tone and movement, and in particular the hypertonia in the extensor muscle groups leading to retrocollis and opisthotonus, relate to contemporaneous injury localized to the globus pallidus and subthalamic nucleus. So this is an extrapyramidal rather than a corticospinal injury. And importantly as well, very early on, during advanced stages of acute bilirubin encephalopathy, aberrations in the auditory brainstem evoked responses are seen with preservation of low acoustic emissions and cochlear microphonics, but involvement obviously of the auditory nucleus and auditory pathway, central auditory pathway. Similarly, the Kernicterus facies, described by Tina Slusher, Dr. Owa, Mike Painter, and Steve Shapiro, also reflects this localized injury. The setting sun sign and paresis of vertical gaze, indicative of ocular motor nucleus involvement. This coupled with eyelid retraction, what's referred to as Collier sign, indicative of dorsal midbrain involvement. And indeed, even the facial dystonia that leads to the stunned, anxious appearance in the newborn reflects globus pallidus and subthalamic nucleus involvement. So in addition to these early phenotypes, the classic long-term Kernicteric sequelae also are entirely consistent with this selective involvement in terms of chorioathetosis and truncal hypotonia, the globus pallidus and subthalamic nucleus, ataxia with cerebellum, again impaired vertical gaze with auditory with the ocular motor nucleus, and then ultimately the appearance of the auditory neuropathy, a spectrum disorder or auditory dyssynchrony, involvement of the auditory nuclei. So again, we have this very, very distinct, highly localized pattern that's evident clinically, both early and later on, and both in terms of the motor and auditory systems. Similarly, the regional topography of bilirubin-induced injury is evident on magnetic resonance neuroimaging. And here we see on the top panel in early T1 weighted neuroimaging, this MRI having been taken on day of life 5 in an infant who evidenced both advanced stages of acute bilirubin encephalopathy during the birth or during the early hospitalization, and later found to have the chronic long-term sequelae of Kernicterus. Early on, this infant shows, again, classic involvement in terms of hyper intensity and the signals on T1 and the globus pallidus and subthalamic nucleus bilaterally. And the same infant 6 months later on T2 weighted MRI, again, showing evidence of the globus pallidus bilaterally and subthalamic nucleus. So the neuroimaging is entirely consistent as well, both early on and later. And the neurocellular pathology also evidenced the same selective neuroanatomic involvement. Again, the injury being primarily subcortical and neuronal. And again, the same regions that we've detailed previously being seen as affected in at postmortem. I did want to just interject here a second in terms of that the literature on neuropathology of Kernicterus can be very confusing if one's not very careful, and is defined, Kernicterus at path is defined as the presence of both the retention of bilirubin staining following formalin fixation and cytopathologic changes of damage in these affected regions. So diffuse yellow staining in the absence of cytopathology is not Kernicterus. This is a controversy that was visited and settled actually decades ago. And I would urge folks who are really interested in this topic to become very familiar with the classic work of Mamdouha Adha-Barmada, her early work in conjunction with John Lucy, and in particular her a chapter on the neuropathology of Kernicterus definitions and debate. It was published in 2000. These are really important seminal contributions to the literature. And even Schmorl in 1903 highlighted these important distinctions in terms of Kernicterus diagnosis. One of the interesting things that's quite evident in terms of the topography of Kernicterus at postmortem is that the pattern is very distinct from that seen in hypoxic ischemic injury. And again, it's a subcortical lesion, does not involve periventricular or cortical white matter. And this table from one of Dr. Barmada's reviews shows the comparative neuropathology of Kernicterus and what was referred to, this is back in 1984, as anoxic ischemic encephalopathy, but currently we would tend to refer to it as hypoxic ischemic encephalopathy or just neonatal encephalopathy. But it's important that with regards to the distinction in terms of the topography of lesions between Kernicterus and HIE, with absent involvement of the cerebral cortex as opposed to cerebral cortex injury in HIE. Again, absent periventricular white matter involvement in Kernicterus, present in HIE. When it comes to the corpus striatum, which refers to the basal ganglia, we're talking about involvement of the globus pallidus in Kernicterus as opposed to the putamen and caudate nuclei in HIE. With regards to the thalamus, it's the subthalamus, subthalamic nuclei in Kernicterus, as opposed to anterior and lateral nuclei in HIE. With regards to Ammon's horn, which is the hippocampus, we're talking about the metabolic resistance sector involvement in Kernicterus, other regions in HIE. And not surprisingly, this distinction in terms of topography is noted on magnetic resonance imaging here in T2 weighted imaging. Kernicterus again with bilateral involvement of the globus pallidus, hypoxic ischemic encephalopathy, involvement of the thalamus and putamen. So if you see an infant like this, this is a MRI on an infant. This is a 4-month-old former 26-week gestation newborn. So this is the T2 weighted MRI. And you see, I don't know if you can follow my arrow, but you see the increased fluid in the extraaxial spaces, this ventriculomegaly ex vacuo, and thinning of the corpus callosum. Now, this is a white matter injury, cortical white matter injury. This is not Kernicterus, okay? But when you see these changes in conjunction with increased bilateral signal intensity in the globus pallidus bilaterally here, this is what Steve Shapiro and his colleagues would refer to as Kernicterus plus disorder. That is Kernicterus plus some other insult that led to the periventricular white matter injury, periventricular leukomalacia, white matter volume loss. And this could be concurrent hypoxic ischemic encephalopathy. It could also be some response to systemic inflammation, as we see in a fetal inflammatory response syndrome in response to maternal cortical amnionitis, and also postnatally with the occurrence of sepsis and necrotizing enterocolitis. But again, important distinction between Kernicterus in terms of the topography and hypoxic ischemic encephalopathy. Now, what accounts for the selective, for these differences? And Michael Johnston at Hopkins at Kennedy Krieger Institute has some really nice papers on this topic. And he highlights the selective injury to the putamen, thalamus, and the cortex from near total asphyxia in term infants is related to excessive activation of the neuronal NMDA and AMPA-type glutamate receptors, a result of mitochondrial energy failure and passive opening of the NMDA channels with activation of these glutamate-containing excitatory circuits. And because of this, and because of where the globus pallidus and subthalamic nucleus are situated in this neural circuitry, the globus pallidus and subthalamic nucleus are spared in HIE because they're downstream to GABAergic inhibitory synapses. And this actually makes a lot of sense, and accounts for why the globus pallidus and subthalamic nucleus may be spared in HIE, but doesn't fully explain why the globus pallidus and subthalamic nucleus are involved in Kernicterus. And this again is a question that remains unanswered, and really necessitates that we think a little bit outside the box and analyze the topography from varied perspectives. And one approach is to identify other conditions that evidence a similar topography, and in particular with regards to the globus pallidus involvement. And there are a host of different such conditions, including, excuse me, carbon monoxide poisoning, cyanide poisoning, manganese toxicity, a host of congenital mitochondrial encephalopathies, including isovaleric acidemia, methylmalonic acidemia, and pyruvate dehydrogenase deficiency. This is an MRI of an infant with pyruvate dehydrogenase deficiency, again showing on this T2 weighted imaging hyperintensity in globus pallidus bilaterally. Indeed, cyanide, manganese, and carbon monoxide are commonly reported toxins associated with basal ganglia lesions. And notably, they share interference with mitochondrial complexes, cytochromes, the oxidative respiratory chain, and mitochondria. And the evidence T2 weighted abnormalities in the basal ganglia. Indeed, as you're all well aware, mitochondria have long been thought a potential bilirubin target, and are of course responsible for ATP generation, calcium uptake and storage, and generation and detoxification of reactive oxygen species. And as shown in the panel above here, you can see consistent involvement of the globus pallidus, but also in some of the conditions in carbon monoxide and cyanide poisoning involvement, as well as other basal ganglia regions, including the caudate and putamen, and also potential white matter lesions. And not all the mitochondrial encephalopathies affect the globus pallidus and subthalamic nucleus. Some instead adversely impact the caudate and putamen, including glutaric aciduria and propionic acidemia. It's also distinctly unusual to see reports of auditory involvement in terms of auditory neuropathy spectrum disorder in conjunction with mitochondrial disorders or carbon monoxide poisoning. There is a syndrome of deafness and dystonia that's been reported, but this is exceedingly rare. It is not of neonatal onset, it's later onset, and it is an etiologically heterogeneous disorder. So accounting for the selective vulnerability of the globus pallidus and subthalamic nucleus and bilirubin-induced brain damage based on mitochondrial targeting and dysfunction remains to be unraveled. Although I would say I think it's still a very tenable explanation and needs much further study. Well, another area of speculation with regards to this topographic pattern reflects regional differences in membrane lipid content or lipid processing. And indeed, there's been a long-standing interest in bilirubin as a lipophilic substance and its ability to partition within the lipid bilayer of cellular and subcellular membranes, including the plasma membrane, endoplasmic reticulum, and our friend here, the mitochondria. And indeed, Cynthia Baer has been doing some very interesting work on bilirubin binding to lipid rafts and the potential role that may play in the pathogenesis of bilirubin-induced injury. And we look forward for the results of her ongoing studies. One of the things that has intrigued me clinically is the overlap of early T1 signal intensity of the globus pallidus and subthalamic nucleus of healthy neonates and those of infants with Kernicterus. And that's shown here in these two panels. On the top panel here, on the far left, again, we have the neonate we described earlier that had both advanced stages of acute bilirubin encephalopathy and ultimately Kernicterus. Again, this is the early, though, T1 weighted imaging. So the first few days to a week or so of life. And on the right, we see two different newborns who had evidence of early stages of acute bilirubin encephalopathy, but not advanced stages, and who underwent aggressive treatment, either with intensive phototherapy or exchange transfusion, and who passed their auditory brainstem evoked responses during the early hospitalization and looked normal neurologically. But based on these MRIs, the pediatric neuroradiologist raised concerns that maybe these infants have Kernicterus. In fact, none of them had Kernicterus. They were perfectly normal on follow-up, both motorically and with regards to their hearing. But you still see hyperintensity in the subthalamic nucleus and suggestion in the globus pallidus in these infants. And on the bottom panel here, again, it's the same baby here with Kernicterus. And here are 4 different babies, all of whom were perfectly normal. In fact, none had any significant hyperbilirubinemia, and none showed any evidence of acute bilirubin encephalopathy. And yet we see hyperintensity in terms of T1 signals in the globus pallidus and subthalamic nucleus. So what we're seeing here as a high T1 signal is subcortical myelin. At least that's the most likely explanation. And due to its high lipid content. Indeed, the T1 signal is a lipid-laden signal. Such that myelination is a key differential in high T1 signal intensity in neonates. And myelin is present in both subcortical gray, and importantly, subcortical gray, as well as subcortical white matter. And indeed, we don't see really any hyperintensity in the paraventricular white matter on these early T1 weighted imaging. So at term postmenstrual age, so 40 weeks, myelin is normally present in specific tracts in the brainstem, including gray matter nuclei, globus pallidus, subthalamic nucleus, dentate nucleus of the cerebellum. So it's easy to potentially misattribute high T1 signal intensity associated with active normal myelination in term infants with Kernicterus. But the question that's always been in my mind thinking about this, is this overlap coincidental, or does it reflect a predilection of bilirubin-induced toxicity to regions undergoing active myelination? Well, Dora Britis and colleagues published a relatively recent paper that suggests that oligodendrocytes are a target for bilirubin-induced injury, and demonstrated marked reductions in active subcortical and cerebellar myelination in the UGT1A1 knockout humanized mouse model of Kernicterus of Bob Tukey and colleagues at UC San Diego. And in this figure, you can see that the percentage of myelinated fibers is significantly decreased in the cerebellum, medulla, and pons in the Kernicteric pups as opposed to their control counterparts. And there was no significant effect on the corpus callosum, which again is a cortical white matter structure. So these are interesting findings along the lines of the importance of myelination. And we certainly know from older literature that bilirubin does bind to myelin. So I think, in particular, this neuroimaging data speaks to the potential of actually advance the neuroimaging beyond the conventional structural MRI that I've been detailing here in terms of T1 and T2 weighted imaging, to explore the topography of bilirubin-induced injury in human neonates, and perhaps even more for the colleagues in Trieste and those doing more basic work, preclinical murine models to look at the bilirubin's effects on neural circuitry and neural networks. Indeed, what's referred to as the diffusion tensor imaging, which is a subset of diffusion weighted imaging with tractography using parent diffusion coefficient mapping, may be very useful in looking at the short and long-term changes along the anatomical pathways. Steve Shapiro would tell you, it's increasingly recognized that movement disorders arise from abnormalities in neural circuitry rather than localized lesions. And indeed, the subcortical regions involved in Kernicterus, the globus pallidus, the subthalamic nucleus, the dentate, the hippocampus, are all interconnected with each other, as well as other subcortical regions via white matter tracks, including the corticopontocerebellar cortical pathway, the corticostriato-thalamocortical pathway. And surprisingly, it's not known how these pathways are affected in the long term in infants and children with Kernicterus. Absolutely surprising. There has not been a single publication using this advanced neuroimaging techniques in an infant or a child with Kernicterus. So we haven't a clue what's going on there. And not surprisingly, there really are, there's only one report, which I'm going to detail in a second, about what's happening during acute bilirubin encephalopathy and its evolution. But this really offers the potential to provide some interesting insights. Again, let me just share this brief case report. So this is a term infant who had a hazardous total serum bilirubin in an outside hospital. It was greater than the upper limits of their assay range, greater than 30 milligrams per deciliter, so in a hazardous range. Indeed, the bilirubin on admission to our hospital, following a period of intensive treatment and hydration, was 28.7. And this was in the context of previously unrecognized anti-big E, anti-little c, hemolytic disease of the newborn. Both at the outside hospital and on presentation or admission to our hospital showed advanced stages of acute bilirubin encephalopathy, opisthotonous retrocollis apnea requiring mechanical ventilation, and a bilirubin-induced neurologic dysfunction score of 9 out of 9. The infant also was noted to be hypernatremic, to have seizures, and so underwent an MRI, including magnetic resonance angiography and venography, to rule out cerebral venous sinus thrombosis immediately following a double volume exchange transfusion. And I'm sorry, there's a lot of detail on this slide. I would urge you to look more closely at the MRI findings in the actual case report. But what was seen is on diffusion weighted imaging was acute diffusion restriction, which is dark signals we see here. It reflects injury to selected white matter tracks from the dentate nucleus through the superior cerebellar peduncles and to the superior thalamic radiations, in addition to T1 weighted hyperintensity in the subthalamic nucleus and globus pallidus. This is a heretofore unrecognized anatomical substrate associated with the regional neuropathologic changes observed in Kernicterus. And whether this whether this ADC map reflects direct injury and or transsynaptic changes along the dentato-rubro-thalamocortical pathway during acute bilirubin encephalopathy is not clear, but it does highlight that bilirubin can have adverse effects on neural circuitry. And again, as I said earlier, as a tool to explore the pathogenesis and the neuropathology and the topography of Kernicterus, advanced neuroimaging is really an untapped resource. And importantly, again, as applied to murine models, would allow one to look serially across time in the same animal as acute bilirubin encephalopathy develops and evolves. Indeed, small animal neuroimaging has made great strides over the past 10 to 15 years with much stronger magnets. It certainly requires, of course, the collaboration of experts in advanced neuroimaging, and not every institution has such devices available. But when they are available, or collaborating long distance, I think holds great potential. Again, getting way beyond just a structural T1, T2 weighted MRI, but to functional MRI, magnetic resonance spectroscopy as well has made huge strides. and positron emission tomography as well. Well, let me shift gears here briefly to the importance of timing, the temporal sequence of bilirubin-induced injury. I think it's important to remember that essentially all the neuroimaging data we have on Kernicterus in human neonates is obtained well after the insult. The case report that I detailed was distinctly unusual to have a exchange transfusion. I'm sorry, to have a MRI done so quickly following an exchange. Most, in fact, are at least 48 to 72 hours in terms of the early T1 weighted imaging during early hospitalization following an exchange. And the later positive the T2 weighted imaging, those take several weeks to months to become evident, but are the true hallmark of Kernicterus. But again, well past the initial insult. Obviously, all the neuropath on Kernicterus in humans is often well after the bilirubin insult, and typically at least 12 to 24 hours following death. And if you look here at this table for one of Parmada's reviews, in terms of the neuropathological findings of Kernicterus, the early changes are at least 2 to 5 days following the insult. So we don't have this is a relatively long interval between the toxic and bilirubin exposure. And we know that bilirubin neurotoxicity can occur quickly following a hazardous exposure. And this is some work from Steve and his group on profiling minocycline neuroprotection, bilirubin-induced adverse effects on the auditory system. But what's important here is that he demonstrated profound bilirubin-induced dysfunction within 6 hours of inducing acute bilirubin encephalopathy in the gun rat pups following sulfa digoxine administration, and included at 6 hours, within 6 hours, complete absence of the auditory brainstem evoked responses. And observed significant dysfunction as early as 120 minutes post-sulfa dosing. Prophylactic minocycline prevents this dysfunction and injury, but minocycline given 120 minutes post-sulfa provides only partial, incomplete protection. So this is a model that offers the potential to explore very early proximal bilirubin-induced events and the evolution of bilirubin neurotoxicity within a very narrow window, clearly defined early window, 6 hours in vivo. You might say, well, there's a wealth of in vitro studies that demonstrate, myriad early adverse bilirubin effects in cell lines within hours, obviously to days of the exposure. And indeed, the breadth and number of the biochemical and cellular effects is wide-ranging and appears to be almost ever expanding. And this is simply a table to outline some of those effects and potential mechanisms underlying bilirubin neurotoxicity. I'm not going to walk through these. Many of you are far more familiar with these details than am I. And these are indeed important studies, but they're defined by the in vitro conditions that they were performed in, and may or may not mirror those that exist in vivo. And in fact, the myriad number of effects has raised questions as to whether bilirubin may be a promiscuous inhibitor. Tony McDonough raised this concern, and actually Thor Hansen, Ron Wong, and David Stevenson also highlight this in their recent physiologic reviews on bilirubin-induced injury. What are promiscuous inhibitors? Well, they're compounds that show nonspecific, indiscriminate, or unselective inhibition protein receptor targets, and they came to the fore with the development of automated in vitro screening for new pharmacologic agents, during which some compounds that showed very strong in vitro activity evidenced no drug-like activity when studied in vivo. And notably, these compounds were typically characterized by high hydrophobicity, high molecular flexibility, and the ability to form microaggregates, all properties shared by bilirubin. And it's unclear what the actual solubility of bilirubin is at body temperature in the protein-free aqueous phase of the central nervous system, full of other solutes. And it may or may not be the same as examined in vitro. Probably not. Very difficult to replicate perfectly in vitro. But it's also known that it's easy to supersaturate aqueous solutions in vitro at room temperature, a condition that is common in many in vitro reports. So it becomes critically important to confirm putative adverse bilirubin effects identified in vitro and interventions to attenuate those effects with studies in vivo. And let me just highlight one study that we performed years ago. I know most of you are familiar with this, but I'll tell it again anyhow, because I think the lesson, at least I learned from this, is how careful we have to be in terms of extrapolating in vitro findings to the in vivo experience and what actually ultimately happens perhaps in babies. We all know that hazardous bilirubin exposures will lead to oxidative stress. It's been repeatedly shown by many people. But is this a mechanism that results in CNS injury and acute bilirubin encephalopathy and Kernicterus? There are a whole host of antioxidants that have been tested in terms of their neuroprotective effects, including taurouracil deoxycholic acid, or TUDCA, or bile acid derivative, shown to be protective against neuronal cells in vitro in terms of bilirubin's adverse effects. We were interested in exploring not just TUDCA, but minocycline, which I must say is the only substance I'm aware of that literally every time it's given prophylactically, gun rat pups do not develop any evidence of bilirubin-induced injury or encephalopathy. And a derivative of minocycline called PMIN, which importantly is a non-calcium chelating derivative. We were interested in this because we thought, well, this might be something that could potentially be translatable way down the line clinically, since this would not be associated, given its inability to chelate calcium, with the adverse effects on dentition, which is one of the main reasons minocycline cannot be used in newborns. We thought to examine this in vivo using the sulfa-induced acute bilirubin encephalopathy approach in the gun rat model. And we looked at bind scores. Again, this is a bilirubin-induced neuromotor index with the maximum score of 5 as a proxy of acute bilirubin encephalopathy severity, brain bilirubin content, and we looked specifically at lipid peroxidation. Not all potential effects of oxidative stress, but lipid peroxidation in terms of 4-hydroxy non-nail or 4-HNE adducts. What we saw here in the top left is that indeed sulfa-induced acute bilirubin encephalopathy in the JJ pup was associated with a significant increase in 4-HNE adducts, and that TUDCA, PMIN, and minocycline, every one of them was associated with significant pre-treatment, was associated with a significant reduction in the cerebellar 4-HNE to levels that actually were comparable to that in the non-jaundiced NJ saline-treated pups. But notably, despite this significant reduction in the 4-HNE by TUDCA, PMIN, and minocycline, only minocycline was neuroprotective against acute bilirubin encephalopathy. The TUDCA-treated pups, the PMIN-treated pups, all looked just like the sulfa-treated pups alone. And again, only minocycline prevented acute bilirubin encephalopathy. So it appeared that lipid peroxidation, this is cerebellar lipid peroxidation, alone was not sufficient to prevent acute bilirubin encephalopathy, which raised questions about the causal link between oxidative stress releases mediated via lipid peroxidation and the genesis of acute bilirubin encephalopathy. So we speculated that the neuroprotective efficacy of minocycline involved actions independent of or in addition to its antioxidant effects. But the main message here is that we need to be very cautious about inferring a causal link between a bilirubin-induced effect and preventing that effect and the development of bilirubin neurotoxicity. In other words, here we had an example of a true, but unrelated. It was true there was lipid peroxidation. Yes, it was true we could prevent lipid peroxidation, but it didn't prevent bilirubin neurotoxicity. So in summary, I think it's important to know what drives the distinctly highly regional topography of Kernicterus as part of providing a more complete understanding of Kernicterus pathogenesis. And I think holds tremendous promise to provide important insights regarding this disorder. And the study of topography goes hand in hand, I think, with identifying these very early proximal events in the CNS bilirubin injury cascade. And we have the tools moving forward for major empirical advance. The transgenic mirroring models that you have in Trieste, that Bob Tukey is utilizing in UC San Diego, and just the good old gun rat model are available, obviously, for a preclinical translational study. But I think the advanced neuroimaging is really the untapped resource that will allow us to explore more fully the regional nature and early events in real time and in sequence, and really holds great potential to be the means to move the field forward. I think it's also important investigators increasingly interpret, integrate, and test their findings in the broader context of trying to explain this the regional topography, and at the very least confirm the findings at the molecular and cellular levels in vivo. And I think we actually hopefully will promote greater discussion of the data integration and explore and test, develop explanatory models of Kernicterus topography. They may even hold potential relevance to other CNS disorders. And with that, I'll close and simply say I look forward to a time when we can get together again face to face in Trieste, and wish all of you safe and healthy holiday season and 2022, and certainly entertain any questions or comments you have. Thank you.
Thank you to you. I mean, that's excellent speech. Very, very inciting, actually. And the bottom line is that we still don't know how bilirubin is getting damaged. Is getting damage to neurons or astrocytes or any other cell, which makes the whole thing even more challenging. There's any question or comments, please?
I have a comment or a question.
Okay.
I guess I kind of like this idea of bilirubin being promiscuous, in that it doesn't seem to have a single protein target, but a range of targets. And I think that's a lot like fetal alcohol syndrome. And I don't know if you've looked at comparisons between, Kernicterus and fetal alcohol syndrome. The timing of the exposure is different. Obviously, for fetal alcohol syndrome, you're exposed in utero rather than, right after delivery. But I wonder if there's some similarities there.
Yeah, that's a great question. And no, I'm not familiar in detail with the specific lesions in fetal alcohol syndrome, although my recollection, and you can correct me if I'm wrong, is there is a substantial cortical involvement in fetal alcohol syndrome. But No, that certainly merits looking at closely. Absolutely.
I think the cortical involvement has to do with the timing of the exposure.
Okay.
So it would be difficult to look at human, models of both, but very relevant, I think, to look at a lot of the fetal alcohol models are just a postnatal rodent exposure. And it would be interesting to compare those models to, like the bilirubin animal models, where you can control the timing of exposure.
Yep. Yeah, I think as I think is clear, hopefully from the presentation, is one of the really unique aspects of bilirubin-induced injury. It is really is a subcortical insult, and one of my points of emphasis was to in fact make that distinction, because there is, at least in my reading of the literature, some confusion or confusing findings, and although the bilirubin may very well affect white matter, it's subcortical white matter, and I just think that's an important distinction to make, and maybe a clue. Why bilirubin doesn't affect the cerebral cortex. But thank you, Cynthia. Appreciate that.
Any other question? Well, no, let me make a comment, general comment. And I think that what we still don't know is whether the damage, different damage at different levels. Let's take into consideration one typical example, the superiors and the inferior colliculi. They're both very similar in terms of vascularization. They receive the same amount of blood. They receive the same amount of oxygen. And one is damaged, the inferior colliculum, the other one is not damaged. So the real question is this related to the fact that less bilirubin is entering is not damaged region, or more bilirubin is metabolized in the respective region? We still don't know. And I like the idea that bilirubin is not just a single hit on the specific target, but it's much more widespread effect, which is actually true in almost all the biological compounds. I mean, if you look sugar is not hitting anything specific in the cell, but it's actually bringing together a sort of a orchestra. And I think that what we need to do is to realize, studying the in vitro, and I like the idea to have things measure in vitro, confirm in vivo, because otherwise it's very hard to make a comparison and make something reported to be harmful or protective useful in the clinical scenario. And it's still a long way to go. I mean, that's fascinating.
Yeah, no, I rightly or wrongly still generally think that the bilirubin exposure, I'm assuming that the bilirubin exposure is pretty much the same everywhere you go in the brain. That may or may not be true. We both have been keen interest in bilirubin uptake and clearance in the central nervous system, and whether there's a regional nature to that phenomenon is unclear. But I guess my thinking is, at least for now, is that initial exposure is the same everywhere. And
any other question?
And then try to tease things out from there. Can I ask you a quick question? Do you in Trieste, do you have anybody or facilities that would permit to advanced neuroimaging of small animal models?
No, unfortunately.
Nobody. Okay.
Nobody.
Yeah.
But actually, we can think about to bring our rats in a facility where they should be available. I think in Padova, they have a large facility. For sure in Bologna, they have a huge facility for MRI and PET scan in small animals and see what we can do. Because this could be, and what we expect to do is if we confirm the most severe phenotype in our GAN rat colony, this could be something that could provide some light on what we're dealing with.
Yeah, I mean, I've just really been struck, and that's why I made it a point of emphasis, is that even in the clinical arena, it is, to me, astounding, appalling, actually. There has not been any advanced neuroimaging, including tractography, in an infant or a child with Kernicterus.
Agree.
Period. That astounds me.
But there's even one thing which is even more challenging, is why in the clinical scenario, babies with equal amount of bilirubin get damaged and others with the same bilirubin get not damaged. So I mean, it's still open. I mean, it's a sort of ongoing research. So the future is yellow.
Yes.
Imagine.
Okay. So if there's no further question, thank you very much, John,
Yes.
for your
Thank you.
excellent talk. And Merry Christmas and Happy New Year to all of you.
Merry
And,
Christmas.
You too. Great.
okay,
Thanks so
thank you
much.
very much. Bye-bye.
Happy holidays.
Thank you, John. Happy holiday. Bye-bye.